Hello students, welcome back to my channel Biomania. I am your biology teacher and once again I am here with some exciting knowledge and some good topics for you. As I have told you in earlier video that I have started a new unit, that is the genetics. Genetics is the very important, very crucial and a little bit tough unit for some students, you can say. So watch these videos very sincerely and also follow the notes which I am providing in my blog, and the link is also provided with the video, so you can get all the knowledge, all the question papers, all the notes at one place. So watch these videos very sincerely. In today's video I am going to cover few important things. Let's have a look about that. First we will study about the Mendel, his birth year, his death year, how he was working with the pre-plants, each and everything we will study. After that we will see the reasons why Mendel work was not recognizable for such a long period. What were the reasons? We will study all these reasons, and after that we will study who were those scientists who rediscovered Mendel's work after a very long period of time, and then after we will study why we are till date following Mendel's law, each and every point which Mendel told us at that time, why we are following that till date. And then after we will study a very crucial point, that is about the pea plant, why he has selected only the pea plant, not any other plant just like rose, tulip or any kind of plant, why he has selected the pea plant. And we will study about the 7 traits, 7 contrasting characters which are selected by the Mendel's also in the end of the video. So watch the complete video very sincerely and make sure you are writing each and every important point and each and every important lines, so that you will not get bothered at the time of your examination. So are you ready for this fabulous journey, guys? Okay, let's start with the information related with the Mendel. When we talk about genetics, the first thing which comes in our mind, that is that who is the father of genetics? Yes, my dear friends, Gregor Johann Mendel is known as the father of genetics. He is the first one who worked on the genetics. He provided us the very first things at that time which we are following till date. Mendel was born on 22nd July 1822 and he worked for 7 years, such a long period as it is. 7 years he worked on Pisum sativum, that is the pea plant, and he died in year 1884. But in this duration his work was not recognizable. After the 16 years of his death, 3 great scientists in 1900 rediscovered Mendel's work. And who were they? They were Hugo de Vries and Correns and Schemack. These were the 3 scientists who rediscovered Mendel's work after such a long period of 16 years. So we have to remember their name because they are very important from the point of view of your exam, that who were the scientists who rediscovered Mendel's work again, or you can say who rediscovered the Mendel's work and in which year they discovered. Now let's have a look about the reasons why Mendel's work was not recognizable for such a long period. Yes guys, that 16-year period is a very long time span. So there are some important reasons, because at that time the first reason you can say that his work was not published properly. It was not published in any international journal so that all scientists of the whole world know about his work. If it was published, it was only in local papers and local journals, so only the local area persons will know about that. So due to the lack of this type of publishing, so his work was hidden by some great scientists. And the most important point, that Mendel was a mathematician and he was working as a priest in a church, and because of that reason, that he is a mathematician, and biologists were not ready at that time to understand his theory, that a mathematician, how can he give a theory about a plant, or because he was a mathematician. And at that time, one more important theory was on the floor, on the top floor you can say, and it was the Darwin's theory. It was the new at that time, and everybody is very curiously searching on that theory, and they are taking too much interest on his theory. So nobody is serious about Mendel's work. So these are the two main reasons why Mendel's work was not recognizable for such a long period. After that, he described factors nowadays which we call as genes. Factors were at that time not acceptable, but at that time Mendel was telling that there is something unit which is transferring from one generation to the next generation, or it is carrying the character from the previous generation and it is passing to the next generation, and nobody is ready at that time to understand this theory, what was that, and that was completely hypothesis. And at that time actually the microscopes and this type of work was in a very elementary stage, so they don't have any kind of proof, they don't know about the DNA, they don't know about these things, so they are not ready to accept that factor thing, that what is that factor, and they are not ready to accept that. So that is also an important reason for his not recognizing his work at that time. Now we will see what are the reasons for the success of Mendel's work. The first one is the selection of the contrasting character. Why he has chosen the contrasting character? Because if we talk about, suppose that if we talk about a tall one, according to me a 5'7" height person is tall, according to somebody else it is 7 feet height person is tall, according to somebody else 7.5. So it can be different for different one. So he has decided to take the all contrasting character. One is the tall and second one is the dwarf. So he has taken height as a contrasting character, tall and dwarf. Tall means tall and dwarf means dwarf. Definitely there is a huge difference between them. So, and the next example you can say flower color, pod shape. There are so many characters he has taken. The 7 character he has taken, and which are mentioned here. And you can see that this is, you can see tall, short, seed shape, round and wrinkle. The one is the round and other one is the wrinkle. Seed color, it can be dark green, light green, sea green, it can be anyone. So he has taken the contrasting one. One is the green and the second one is the yellow. If we talk about seed coat color, so he has taken one is the green, not other one is a light green, dark green, sea green or any other kind of green, olive green. He has taken white. So there is a huge difference between them. So these are the all contrasting traits which he has taken. So that is the major reason for his success. Until date we are following these characters. Now the next one is the large sample size. He has chosen pea plant, and in each pot we get so many number of seeds, a minimum 3 to 4 seeds in each pot we get, and a large number of pods we get in a pea plant after the fertilization, or you can say. And that is also a very big reason that he is having a large sample size, so he can do more and more experiments, and so he can provide more and more results. He is not working only a single plant, he is working a large number of plants, so he has so many proofs. So that is also a very big reason to accept his work. Next one is the data analysis. As I have told you that Mendel was a mathematician, so his working style, calculating data, that is a very clear manner. Each and every calculation were very clear and very simple. So his data analysis method is also very important. And next one is that the contrasting traits which are chosen by Mendel, they don't have any kind of linked gene, so there is no any phenomena of linkage, so no use of linkage occur in his work. So that is also a very big reason of his success. So what were the reason of his success? The first one is that he has chosen the contrasting characters. Next one is that he has a large sample of size. It means he is working on so many plants. He has too much results. And his data analysis is very simple and very correct. And then after, the fourth one is that there is no use of linkage. So these are the reason why Mendel work is very successful till date. We are still following as it is, as he has provided at that time. Now let's have a look about the reason why he has selected the pea plant only, not any other plant. Because the lifespan of a pea plant is very small. It's not more than 6 weeks, or you can say not more than 7 to 8 weeks. In 4 to 6 weeks, his complete life cycle will be covered. You will get the seed, and next generation you can start. So in a year you can perform so many experiments. Then after, self-fertilization and cross-fertilization, both are possible in the case of pea plant. Next one is that their seeds, most of the seeds, you can say almost all the seeds, do not show dormancy. They all are viable. So that is also a very good symptoms or very good thing for selecting a pea plants, because he is getting all the viable seeds so that he can perform more and more experiments to get the good results or get a large number of the same result. And number of seeds produced are very high. Yes, definitely, as I have told you that in each pod they get a large number of seeds, and there are so many pods are present in a single pea plant. So the number of seeds or number of results you can say produced is very high. So these are the reasons why he has selected the pea plant. And one more thing, that is the contrasting character. In a particular single plant, he is getting 7 contrasting traits in a single plant. So that is also a very important reason for selecting that particular plant. In other plant maybe he will not get that. He can be, but not that much simple with all these qualities. So that is also a very big reason. So now I think now these points are clear to you, that short lifespan, self and cross fertilization, seeds do not show any kind of dormancy, number of seeds produced is very high, and the contrasting characters. These are the main 5 reasons why Mendel has chosen the pea plant, not anyone else. Now the important thing is that what were the traits, what were the contrasting traits, 7 contrasting traits which Mendel has chosen, and what are they? So you have to learn these traits, clear guys, so that you can perform the crosses perfectly. There will be no any confusion in them. The first one is the stem height. In case of a stem height, there is the dominant one and recessive one. As I have told you in earlier video, the dominant can represent himself in a single allele. Suppose that if we are having capital T and small t, so capital T represent to the dominant one and small t represent to the recessive one. If single capital T is present along with the small t, then it can represent its phenotypic character, not genotypic one, phenotypically. So the phenotypically how it will look tall. But when we talk about the recessive, it can show or it can represent itself only when both alleles are present in the same manner. It means both small t and small t are present, only then it can represent its phenotypic character. Then after only it can be a short plant or dwarf plant, you can say. Clear guys, what is the dominant and what is the recessive? So have a look about the first one, that is the stem height. The first one is the dominant and the second one is the recessive. These are the contrasting traits, and here you can see that is the tall and that is the dwarf. When we talk about flower color, that is the violet and this one is the white. They both are contrasting, no any other between. Yes, definitely I know codominance is coming in your incomplete dominance or codominance is coming in your mind. We will study that in upcoming videos. So let's focus only in these 7 contrasting traits right now. Now flower position. Now about the flower position, it can be axial, not any other side. It can be axial or it can be terminal. Either it on the top, either it on the side. So that is the contrasting one. Then after pod shaped, either pod will be completely inflated or it can be constricted. It's not completely inflated. Pod color, yes, it's not dark green, light green, olive green, sea green, not like that. It can be green one and it can be the yellow one. Now seed shape, it can be the round or it can be the wrinkled one. Yes, not small round, big round, not like that. It can be round, either it is small, either it is big, doesn't matter. Or it can be wrinkled. Seed color, it can be green or it can be yellow. Here remember that the yellow one is the dominant trait and the green one is the recessive trait. Now I think all these points are clear to you guys, and if you are facing any kind of problem with these topics, you can tell me in comment section so that I can clear your doubts immediately. And about its relevant notes, I am providing link along with the video so you can get the notes and questions related with that topic. Visit my blog also for that. So I think now about the Mendel's information, the reasons why his work was not recognizable, and who were the scientists who rediscovered his work, and what is the reason of Mendel's success, why he has selected the pea plant, and what were the 7 contrasting traits. All these things are clear to you, and still if you feel any doubt, you can tell me. Okay, thank you for watching my channel and for getting latest update and for getting latest update about the upcoming videos. You have to subscribe my channel and also share with your friends so that they will also get benefit from that. So thank you for watching it. Bye bye this time. Meet you soon.